Hello, and welcome to OGT CytoCell University. Today's session, Fish 102, Building on the Basics, is the next offering in our CytoCell educational series. We hope that you find today's information valuable and informative. In the previous CytoCell University session, you learned about key concepts in fish testing. These included a general introduction to fish, requirements to run the assay, some basic abnormal signal patterns, fish interpretations, and even a few troubleshooting tips for tissue and hematologic specimens. In today's module, we will expand on some of those key concepts. We will start by recapping what is fish, its benefits and applications, and then move on to an introduction to fluorescence microscopy. Here, we will review the basic components, discuss how the fluorescence microscope works, filter options, including how to select the correct ones, how oil immersion is used, and the purpose of fish counterstain. Then, we will review some chromosome morphology basics, chromomaps, and explain how to establish the 5' and 3' designations for some of OGT's CytoCell fish probes. Next, we will review the different types of fish probes and the design strategies employed for some of them. We will provide examples, review the expected normal and abnormal signal patterns, and offer guidance for analysing these probes. Lastly, we will finish today's module with a brief synopsis of OGT's CytoCell products and support offerings. Before we dive in, it is essential to note that, other than OGT's FDA-cleared CytoCell fish probe kits for AML and MDS, the rest of our fish probes are analyte-specific reagents, or ASRs. Due to the FDA's guidelines for the regulation of ASR products, OGT is extremely limited on the guidance we can provide for our ASR products, as these are considered to be used as building blocks for laboratory-developed tests, or LDTs. Most LDTs in the US are regulated under the CLIA regulations, and labs should always follow the necessary regulations to maintain CLIA compliance. We will be reviewing the differences between FDA-cleared probes and ASRs more in-depth in the CytoCell University Fish 103 session. Stay tuned for its release. Let's begin today's session by briefly reviewing some of the concepts we learned in Fish 101, Basics of Fish. Cytogenetics is the study of chromosomes and involves testing various specimens to identify genetic abnormalities. Fish, or fluorescence in situ hybridization, is an assay within the cytogenetic laboratory's toolkit that utilizes a labeled DNA sequence, known as a probe, to assess a specific region on a chromosome. This technique may be performed on various types of specimen, including prenatal cells from amniocentesis or chorionic villus sampling, constitutional samples from blood or skin, hematologic specimens from blood and bone marrow, or formalin-fixed paraffin-embedded FFPE specimens from solid tumours. Fish is helpful for determining or picking up chromosome alterations, including extra or missing copies of chromosomes, otherwise known as aneuploidy, deletions, duplications, rearrangements, translocations, and amplifications. As you learned during the first session, fish encompasses 4 main steps. In the first step, a fluorescently labelled DNA probe with a complementary DNA sequence for the region of interest is added to the pre-treated slides. A cover slip is then applied to these slides and is sealed with a rubber glue solution. In the second step, the slides are heated to encourage the denaturation process of both the specimen and probe DNA strands. These strands separate, allowing the probe to gain access to the target DNA in the patient sample. In the third step, after the short denaturation, the temperature is reduced which allows the probe to anneal or bind to the target DNA in a process called in situ hybridization. Finally, in the last step after hybridization is completed, the slides are washed, a counterstain is applied, and analysis or interpretation is performed using a fluorescence microscope. Please feel free to review the video from session 1 if you would like a more in-depth description of this process, or contact our support team via the OGT website if you would like a full-size What is Fish poster for your laboratory. As we mentioned in the previous slide, one of the benefits of fish is the accurate detection of an assortment of chromosomal aberrations. Compared to conventional karyotyping, which involves culturing cells and capturing chromosomes in metaphases during mitosis, fish allows its analysts to detect aberrations in both the metaphase and interphase stages of the cell cycle. Additionally, fish provides better detection of minor structural alterations, like microdeletions and cryptic translocations, commonly missed by standard karyotyping. Fish's high sensitivity, specificity, and short turnaround time have made it an essential technique for any cytogenetic laboratory. Furthermore, the fish assay can be used in many applications, for both the research and clinical settings, oncological and constitutional testing, and is performed on a variety of specimen types, including cell suspension and FFPE samples, which is the reason it has become such a routine assay in labs around the world. Now that we have recapped fish's different benefits and applications, let's explore a bit more about fluorescence microscopy. To analyze your fish slides, it is essential to have the following components. First, you will need a phase contrast microscope to examine your prepared slides for quality and cellularity before applying the fish probe. This microscope is usually placed in the specimen preparation room or wet lab area. Once you have examined your slides, you can pre-treat your slides, apply the fish probe, and proceed with the hybridization process. For slide interpretation, you will need, of course, a fluorescence microscope with a variety of filters and the correct objectives with different magnifications. Plan apochromat objectives, for example, are manufactured with different magnifications at 10 times, 60 times, and 100 times. These objectives provide maximum resolving power, color purity, contrast, and image flatness for observation and photomicrography. You will also need an illumination system that emits the white light from the lamp house and the consumable portions of your illumination system, which can either be just lamps, bulbs, or light guides depending on the system. To further amplify the image, you need to apply a small drop of immersion oil and view the slides using the 100 times or oil objective. And lastly, although not a necessary component to view and analyze the slides, many labs have adopted a full imaging system. These systems, which are available from various vendors, can capture and archive fish images. Newer systems can also perform fish scanning and tissue matching. This will be the most expensive capital expenditure for a fish laboratory, and is still considered optional. A fluorescence microscope is an optical microscope that functions on the simple phenomenon of fluorescence. This is when a specimen is illuminated with a specific wavelength of light, which is then absorbed by the fluorophores hybridized during the fish setup process, and then, in turn, emits light of longer wavelengths. That is, light of a different color to the absorbed light. The illuminated light is separated from the much weaker emitted fluorescence through a spectral emission filter. All of this happens using 4 primary components in your fluorescence microscope: a light source, an excitation filter, a dichroic mirror or dichroic beam splitter, and an emission or barrier filter. Analysts may use a fluorescence microscope every day, but do not know how it actually works. Today, we will walk you through this diagram to show you how all the parts work together. When you turn on your fluorescence microscope, white light passes through the excitation filter. This filter allows only the desired excitation wavelength to pass through. In the example shown here, only blue light is emitted that falls within approximately 473 to 498 nanometers. This blue light then reflects off the dichroic mirror, or beam splitter, down toward the sample through the objective. This light then excites the green-labelled or green-emitting fluorophores of the specimen and then travels back up through the objective to the dichroic mirror, which transmits the longer wavelengths toward the eyepieces and the camera. Prior to seeing the light, though, the emission filter blocks the excited light and transmits only the desired fluorescence emission wavelengths to the eyepiece, generating the typical green or FITSI fluorophore that is seen during analysis. This is why fluorescence microscopy requires a very specific set of filters in order to properly view each fluorophore. Filter sets include 3 separate parts: an excitation filter, an emission or barrier filter, and a dichroic mirror or dichroic beam splitter that work together to allow or block the appropriate wavelengths of light required for viewing a specific fluorophore. Filter sets are sold in 2 different options. Firstly, they can be purchased either mounted together into a filter cube specifically designed for the turret of a specific microscope make and model, or secondly, they can be purchased unmounted for use in a filter wheel or for manual replacement into your pre-owned filter cubes. Each of these options, however, has its pros and cons. For instance, while unmounted filters are cheaper to replace, care needs to be taken when installing them. The filter surfaces should not be touched with your fingers, and they require correct positioning according to the light path in the microscope. Also, replacing unmounted filters in a filter wheel may require replacing all the excitation filters at the same time, so all the components will work together properly. On the other hand, mounted filters are easier to replace in the microscope turret and can typically be popped into place or require the simple tightening of a screw. However, these cost more because one pays for the convenience of the filters already being correctly installed into the cube. Either way, replacing filters should be performed by either a laboratory technologist familiar with the fluorescence microscope or a professional microscope technician. Also, remember that filters are considered laboratory consumables. While replacing older filters is recommended every 3 to 4 years, some newer filters are designed with a sputter coating that extends their lifetime. It is always recommended to replace your filters as designated by their manufacturer. Your laboratory may wish to assign one of your laboratory technologists to perform maintenance and check filters periodically for wear and tear, or have them checked by qualified personnel as part of your microscope's preventative maintenance program. Whoever performs it, a degraded filter is typically easy to spot. The filter will have a mottled appearance that starts on the edges and will eventually completely obscure the filter. This will impede an analyst's ability to analyze fish signals correctly. And, as you can see, the image on the top right shows a degraded filter that needs to be replaced. To extend your filter's shelf life and prevent premature burnout, be sure to properly close the shutter when you are not using the microscope to avoid overexposure to UV light. DAPI, red, orange, green, aqua, and gold are among the most common fluorophore colours in fish. Different filters are available to view these fluorophores in either single, dual, or triple bandpass combinations, meaning you can see 1, 2, or up to 3 fluorophores simultaneously. When choosing your microscope filters, it is essential to note that different manufacturers offer probes labelled with various fluorophores, and proper care should always be taken to ensure correct analysis of your laboratory specimens. For several years, the College of American Pathologists, or CAP, has included an item on the All-Common Inspection checklist regarding this issue. In the Instrument and Equipment Maintenance Function Check section, requirement COM 30685 microscopes for fluorescence testing states that the microscopes used for fluorescence testing are used with filters and slides appropriate to and verified in conjunction with the test or tests being performed. The evidence of compliance suggests having a written procedure that specifies the filters being used to analyse a clinical specimen correctly match the probe manufacturer's recommendations. For that purpose, OGT recommends the maximum excitation and emission wavelengths found in this chart to properly view the fluorophores found in CytoCell probes. This chart can also be found on the OGT website at www.ogt.com/US on the Fish Basics resource page. Please note that OGT offers its customers a 30-day free-of-charge filter evaluation on chroma filters, and our Fish FAS team is always available to assist you in choosing the right filter for your lab's needs. Let's review how to choose a filter set on your own. We will use graphs from Chroma's website as an example. When purchasing a new or replacement filter for your microscope, you always want to ensure that the peak spectra transmission of the emission and excitation for your probe manufacturer's fluorophores or fluorochromes, shown here, closely aligns with the bandwidth, shown here, of the excitation and emission specs for the filter you are planning to purchase. This will ensure that the signal intensity and strength for the given fluorophore will be optimal for analysis. If, by chance, you choose the wrong filter, where the excitation and emission spectra do not align with your manufacturer's fluorophore recommendations, you might end up with a situation similar to the one seen in this graph. Here, a red filter was incorrectly chosen to analyse an orange fluorophore. The peak spectra transmissions of the orange fluorophore do not align with the excitation and emission bandwidths of the red filter set. As you can see here, there is a very minimal wavelength of the orange spectra that will be excited using this filter set and will ultimately reduce emission. When reading fish, this will make the signals appear smaller than normal, or possibly even too dim to visualize, and therefore could hinder proper analysis. That is why choosing the filter within the specs that most closely align with your probe manufacturer's fluorophores is recommended. Please note that although the OGT's CytoCell catalogue fish probes are manufactured with a Texas red fluorophore, some of our custom MyProbes options may contain orange. Be sure to always check the product specifications of each probe before use, regardless of vendor, to ensure the optimal signal intensity. Now, let's explore two different examples of fluorescence microscope illumination setups. First, this is an example of a fluorescence microscope with an attached HBO mercury lamp house. In the HBO acronym, the H stands for atomic symbol Hg, meaning mercury. B is the symbol for luminance, and O is the unforced cooling. This lamp house holds a mercury HBO lamp, or short arc mercury bulb, and there is a separate burner control unit that houses the on-off switch for the lamp, which monitors hours used. There are pros and cons to this type of system. Mercury lamps provide intense illumination and are typically more cost-effective. However, these lamps require a significant warm-up period before reading and cannot be switched on and off within short periods of time. Also, each new lamp requires centering or alignment. These lamps typically have a shorter lifetime, somewhere between 200 to 300 hours, and there is always the potential for the bulb to explode. Additionally, there are two key factors to remember with this illumination source. The first is to avoid getting fingerprint oils on the lamp when replacing, as they can cause premature failure. And the second is that the lamp usage counter needs to be manually reset. If the counter on the burner control unit has stopped working, then the laboratory should keep a log of the usage and the total accumulated hours. This is essential because, as noted previously, mercury lamps can be an explosion hazard and could potentially release harmful mercury vapor if used beyond their lifetime. The second type of light source we will discuss is an external illuminator system. This illuminator system houses a high-pressure 120-watt mercury short-arc lamp, or bulb, and can digitally monitor usage. It is connected to the fluorescence microscope by a fiber optic light guide. Compared to the illuminator in the previous slide, this system provides the convenience of the lamp being pre-aligned in the housing, the mercury lamps having a longer life, approximately 2,000 hours worth, and the lamp requiring less warm-up time before reading. One of the disadvantages of the external illuminators is that your lab will have more consumable products to replace, which include both lamps and light guides, and these bulbs still have the potential for explosion. Again, please note that you should not touch the lamp's surface, as fingerprint oils can cause it to prematurely fail. And if your laboratory is using any mercury bulbs, be sure to always dispose of them properly in accordance with your laboratory guidelines. Before leaving this slide, it would be remiss if we did not mention that external illuminators can also be purchased as LED systems. Although these involve a higher upfront cost, these units claim to provide considerable savings over the unit's life, require no warm-up time, and bulbs can last for greater than 10,000 hours. Every fish laboratory has their choice of illumination system options, which can be selected based on their budget and safety concerns. Now let's discuss why we use immersion oil in fish microscopy. As you can see in this image on the left, when oil is applied to the glass, it replaces the air gap and reduces light refraction between the lens and the glass, and therefore increases the resolving power of your microscope. By using the appropriate immersion oil with a high refractive index, fish images appear clearer due to the fact that the numerical aperture of the objective lens has increased. To properly apply the immersion oil, place a small drop of it on the spot of light. Then, slowly rotate the 100 times objective into alignment while checking to ensure it does not strike the slide. If focused correctly in the 60 times objective, the 100 times will rotate into place without striking the slide. Remember that excessive amounts of oil are not required, as just a minimal amount can be used to view an entire fish probe. Also, by using less oil, it will help you to avoid mixing it with the DAPI counterstain, which creates a blue haze that will obscure your signals. Fish microscopy requires the use of a counterstain solution such as DAPI. DAPI works by binding firmly to the DNA's adenine-thymine-rich regions and aids in the visualization of hybridized DNA fish probes by counterstaining all DNA that is present blue. In addition to using DAPI-stained interphase nuclei to find the correct focal plane of the slide for analysis, it also allows the technologist to assess the morphology of the cell. Many manufacturers' DAPI products include antifade, a solution that helps prevent the permanent loss of fluorescent signal quenching that can result from photobleaching or prolonged exposure to high-intensity light sources. All of OGT's CytoCell DAPI products contain antifade and are available in a few concentrations and size options. DES, or DAPI 8th strength, is a component in our FDA-cleared probe kits for AML and MDS and is recommended for use with cell suspensions, or can be utilized for FFPEs. The DES vials supplied in the FDA-cleared probe kits for AML and MDS are 150 microliters. However, this product is also offered for sale individually in 500 microliters, 1000 microliters, or the brand new 10 milliliter sizes. DFS, or DAPI Full Strength, is a stronger concentrated version that some labs prefer to use for FFPE tissue applications. It is offered in either a 500 microliter or the brand new 10 milliliter size. Lastly, CytoCell offers the DSS, or DAPI 16th strength, which is the least concentrated option for labs that prefer a less intense counterstain. It is available in a 500 microliter vial size only. Please reach out to our FAS team through the OGT website if you would like to evaluate any of our DAPI products. In the last session, we briefly reviewed the layout of OGT's CytoCell chromomaps, but let's discuss those a bit more in depth and cover some basic chromosome anatomy in the next 2 slides. Chromosomes are thread-like structures made up of strands of DNA tightly coiled around proteins called histones, which appear as sister chromatids linked by a constricted region called a centromere. The centromere is a highly repetitive DNA sequence of a chromosome that separates the sister chromatids into the arms of the chromosomes, as seen in the image on the left. These arms are the short P arm and the long Q arm. The letter P was chosen to designate the short arm to represent the meaning petite, or small in French. The letter Q was selected as the longer arm solely because it was the next letter in the alphabet. Laboratories commonly use a technique called G-banding, or Giemsa banding, to stain condensed chromosomes after a harvesting procedure to produce a characteristic banding pattern for each chromosome. In G-bands, the light regions will be early replicating, meaning they are euchromatin and GC-rich or gene-rich, whereas the darker regions that absorb more stain will be late replicating, meaning they are heterochromatin and AT-rich or gene-poor. The tips of each chromosome have a repetitive, non-coding nucleotide region called a telomere. Telomeres play an essential role in maintaining chromosomal integrity and tend to shorten during normal cellular aging, and can be involved in constitutional translocations. Besides banding patterns and telomeres, technologists should also note the centromeric position on chromosomes. The image on the right shows 3 major types of chromosomes, based on the position of the centromeres: metacentric, submetacentric, and acrocentric. Metacentric chromosomes, such as chromosome 3 shown here, have their centromere in the centre, resulting in both the P and Q arms of the chromosome being of approximately equal length. Submetacentric chromosomes, on the other hand, have their centromere slightly offset from the centre, as you can see in chromosome 4, which leads to a slight asymmetry in the length of the 2 arms. Acrocentric chromosomes have a centromere which is considerably offset from the centre, leading to a long Q arm and short satellited P arm, such as that in chromosome 13. During analysis, technologists will match and analyse chromosomes according to their banding pattern to look for abnormalities. Once all the chromosomes are analysed, they can be aligned in order of autosomes, chromosomes 1 through 22, and then the 2 sex chromosomes, X and Y, to produce a karyotype. A normal karyotype is 46XX or 46XY. When ordering fish probes, it is essential to understand the format in which they are labelled. OGT's CytoCell chromomaps visually present the ideogram of the targeted chromosome, the cytogenetic banding nomenclature, the probe fluorophores being used, as well as the specific gene coverage and clone size information for the regions of interest in kilobases. In this example chromomap for the FDA-cleared probe kit USA LPH 024 del 5Q deletion, we see the fluorophores represented on a chromosome 5 ideogram at the 550 band level. This probe kit consists of a 186 kilobase probe, labelled in Texas red, covering a region within 5q31.2, including the D5S500 marker, and a 376 kilobase probe, labelled in FITSI green, within 5p15.3, including the D5S630 marker. Chromomaps are available for all OGT's CytoCell catalogue fish probes. In addition to the fluorophore details, the chromomap also includes information on gene orientation to allow the 5' or 3' probe position to be seen in relation to the gene or region of interest. The arrows on the genes indicate the direction of transcription, which always occurs in the 5' to 3' direction. As seen here, those genes with an arrow above, pointing to the right, are located on the DNA plus or sense strand. The sense strand contains the exact nucleotide base sequence as the mRNA and encodes for a functional protein with a thymine base instead of uracil. However, genes that have an arrow below the gene, pointing to the left, are on the minus or antisense strand. The antisense is the non-coding strand of DNA. A cell uses the antisense DNA strand as a template for producing messenger RNA, specifically mRNA, that directs the synthesis of proteins. The main difference between sense and antisense strand is that the sense strand is incapable of being transcribed into mRNA, whereas antisense strand serves as the template for the transcription. Now let's use the FDA-cleared USA LPH 013 MLL, also known as KMT2A break-apart probe, to walk through an example of how to determine which fluorophore provides the 5' or 3' coverage. As you can see here, the arrow location for the MLL, also known as KMT2A gene, is on the top of the strand and pointing to the right, which signifies it is located on the sense or plus strand of the DNA. This means that the 170 kilobase FITSI green probe that is centromeric to the gene is in the 5' position, while the 87 kilobase red probe that is telomeric to the gene is in the 3' position. Now we will move on and review the different types of fish probes, their designs, and how to analyse them. There are 3 different types of fish probes: whole chromosome paints, repetitive sequence probes, and specific sequence probes. Whole chromosome paints are typically used more in research settings, and OGT no longer sells these types of probes. However, we do offer a range of the other 2 types that are more commonly used in the day-to-day setting of the cytogenetics laboratory. We will discuss both of those in the next few slides. There are 2 subtypes of repeat sequence probes, the first of which is seen here: satellite DNA-based enumeration probes, which you may also see referred to as centromere probes, or CEN probes, are chromosome-specific sequences generated from highly repetitive human satellite DNA located in the centromeric, pericentromeric, or heterochromatic regions of each chromosome. These probes are commonly used to detect chromosomal aneuploidies and offer the fish analyst large, bright signals. OGT offers CytoCell satellite enumeration probes in a concentrated 15 microliter format in either red or green fluorophores, as seen here on the right. Or we also have some available in a blue fluorophore, which can be found on our website. The other type of repeat sequence probe targets the subtelomeric regions of chromosomes. As their name implies, subtelomere probes are located in the most distal regions of the chromosome arms. OGT's CytoCell subtelomere-specific probes, or LPT probes, help identify 41 of the 46 human subtelomeres excluding the p-arm subtelomeres of the acrocentric chromosomes. The X and Y chromosome p-arms share the same subtelomere clone, as do the X and Y q-arms, due to the pseudo-autosomal nature of these regions. OGT offers these LPT catalogue probes in a 15 microliter concentrated format and are available in either a red or a green fluorophore. P or Q telomere probes can be ordered individually based on your laboratory's needs. The next type of FISH probe is a specific sequence probe. This type of probe may also be referred to as a unique sequence probe or a locus-specific probe. These probes target a specific gene or region on the chromosome that may be commonly implicated in an aberration for a specific clinical indication. Specific sequence probes are further categorized by the type of aberration detection strategy. The strategies utilize different designs that will pick up deletions and gene rearrangements, including inversions and translocations. Two such examples of these designs are shown here. The one on the left is the del20q deletion probe, and on the right is the CBF-beta CBFB MYH11 translocation dual fusion probe. We will review the different types of specific sequence probe design strategies one by one, providing each probe designs expected signal patterns for both normal and abnormal cells, and the specific analysis guidelines for each probe found in their instructions for use, otherwise known as an IFU. Another type of FISH probe design to be aware of is that of an amplification probe, which will not be covered in today's presentation. First, let's discuss the deletion probe design using an example from one of our FDA-cleared CytoCell FISH probe kits for AML and MDS. The P53 or TP53 deletion probe is designed to detect a deletion of the TP53 gene, or tumour protein P53, that is localized to 17p13. The probe for the region of interest in deletion probes is typically labelled in the red spectrum, as you can see on this chromomap, where a 161 kilobase probe is labelled in Texas red. This probe covers the entire P53 gene, extending 74 kilobase telomeric to the gene and covering a region centromeric to the gene, to just beyond the marker D17S655. A second probe, labelled in FITSI green, covers the chromosome 17 centromere, D17Z1 region, and acts a ploidy control. TP53 deletions have been identified in a wide range of human malignancies. For instance, the loss or deletion of TP53 in acute myeloid leukemia, AML, and myelodysplastic syndrome, MDS, is associated with a poor outcome and is often seen as a marker of disease progression or secondary disease. During analysis, an analyst will expect to see normal cells have this signal pattern on the top right-hand side. There are 2 red and 2 green signals, or 2R, 2G, as it is typically written, reflecting 2 copies of the P53 gene and 2 copies of the 17-sen region. Though, as you can see on the bottom right-hand side, an analyst may also see the expected abnormal signal pattern as well, which will be 1 red and 2 green signals, or 1R, 2G. Please note that no fusion or yellow signals should be recorded when reading this probe. However, other signal patterns are possible in samples that contain aneuploidy or unbalanced rearrangements. When analysing a P53 deletion probe, if ever in doubt as to whether a cell may be analysable or not, an analyst should skip over that cell, and care should be taken to not analyse nuclei that are too close together to determine their boundaries, while also avoiding overlapping nuclei where all the areas of both nuclei are not visible. However, when a well-defined nucleus with bright signals is found, the analyst will want to know how to count these examples seen on the right. First, A cell like this one on the top right may be seen where one of the red signals may appear to be diffuse or spread out. This cell should be analysed as 2 red and 2 green signals. Secondly, cells like the one on the bottom right may also be seen that appear to have a small gap. If this happens, consider the guideline that if 2 signals of the same colour touch each other, or the distance between them is no greater than 1 signal width apart, or if there is a faint strand connecting the 2 signals, then it should be counted as a 1 signal. Therefore, this cell should be analysed as 2 red signals and 2 green signals. Please remember, although red signals were used in these examples, these rules apply to green signals as well. The next specific sequence probe we would like to review with you today is for a gene rearrangement, or break-apart probe, as it is commonly called. This probe strategy is used when you do not necessarily need to know the translocation partner, but only that the gene of interest has been rearranged. Here you can see an example of the MLL, also known as KMT2A break-apart probe, from OGT's FDA-cleared CytoCell fish probe kits for AML and MDS. The chromo map on the left shows an 87 kilobase probe, labelled in Texas red, covering a region telomeric to the MLL, also known as KMT2A gene, including the marker SHGC 111513, and a FITC green probe covering a 170 kilobase region centromeric to the MLL, also known as KMT2A gene, spanning the CD3G and UBE4A genes. The KMT2A gene encodes for a histone methyltransferase, which functions as an epigenetic regulator of transcription, and rearrangements are frequently reported in patients with AML and in patients with therapy-related MDS, albeit at a lower frequency. Historically, KMT2A rearrangements in acute leukemia were associated with a poor outcome. But recent studies have shown that the prognosis is highly dependent on the fusion partner and may differ between children and adults. There have been over 80 different KMT2A partners described to date, but some of the common genes are AFF1, ELL, MLLT1, MLLT3, MLLT4, and MLLT10. When analysing a break-apart probe, normal cells will have the expected signal pattern of 2 red, green, yellow, or fusion signals, seen here on the top right, typically designated as 2F, reflecting 2 normal copies of the MLL gene on each chromosome 11. An example of the expected abnormal signal pattern seen with this probe is here on the bottom right, in which the analyst will see 1 red, 1 green, and 1 fusion, or 1R, 1G, 1F. The separation or gap between the single red and green signal should be greater than 2 signal widths apart. Please note that there also might be other signal patterns in both aneuploidy and unbalanced specimens, and because this is a break-apart probe, it cannot be used to determine the rearrangement partner. The guidelines for analysing the MLL, also known as KMT2A break-apart probe, also include instructions that analysts should be sure not to analyse nuclei clumped together, where cell boundaries cannot be determined, and areas that have overlapped nuclei where all of the nuclei is not visible. However, an analyst will need to pay attention to the signal gap between the red and green. Fusion signals can either fluoresce as yellow or be observed as red and green signals in very close proximity to one another. An example of this can be seen here, where if an analyst sees a gap between the red and green that is less than 2 signal widths, they should score the red-green as a fusion. Or, when an analyst sees a diffuse signal with a strand connecting the 2 signals, it should also be scored as a fusion. All technologists should be trained to interpret a fusion and break apart in the same manner, in order to ensure the highest concordance of results between analysts. The third type of specific sequence is a tricolor break-apart probe strategy. Although it is similar to the dual-color break-apart, a tricolor design can be used to detect two different types of rearrangements, which can occur on the same chromosome. For example, OGT has the only FDA-cleared probe design for an EVI1, also known as MECOM break-apart, that will detect both a gene inversion by exhibiting one signal pattern and a translocation by exhibiting another signal pattern. This chromomap shows OGT's CytoCell FDA-cleared EVI1, also known as MECOM break-apart probe kit, consisting of a 158 kilobase probe, labelled in Texas red, telomeric to the D3S4415 marker, and including the LRRC34 gene, a 181 kilobase FITC green probe covering the centromeric portion MECOM gene, and beyond marker D3S1282, and a blue probe that covers a 563 kilobase region centromeric to the MECOM gene, including the D3S1614 marker. MECOM encodes a zinc finger protein that is inappropriately expressed in the leukemic cells of AML and MDS patients. This deregulated expression is often due to chromosomal rearrangement involving 3q26.2, with the two most common aberrations being the translocation 33q21q26.2 and inversion 3q21q26.2. The breakpoints for the translocations and inversions vary considerably. Inversion breakpoints are found centromeric too and include the MECOM gene, covering about 600 kilobase, while the majority of breakpoints in 3q26.2 translocations are telomeric to the MECOM gene and cover a region including the telomeric end of the MDS1 gene and the MYNN gene. An aberrant expression of the MECOM gene has an unfavourable prognosis and, potentially, a very aggressive clinical course. OGT has designed this CytoCell probe to exhibit two red, green, blue fusions, or 2RGB, when a normal signal pattern is observed, reflecting two copies of a normal MECOM gene. However, because of this probe's design, there are two potential abnormal signal patterns that analysts may observe. First, a cell that harbours a translocation 33 will exhibit one red, one green-blue fusion, one red-green-blue fusion, or 1R, 1GB, 1RGB signal pattern. Second, for a cell that harbours an inversion 3q21q26.2, the analyst will observe an abnormal pattern that will appear as one red-green fusion, one separate blue signal, and one red-green-blue fusion signal, or 1RG, 1B, 1RGB. Since MECOM has also been shown to be rearranged or translocated with other genes, including RUNX1 on chromosome 21 in therapy-related disease via the translocation 321q26.2q22 translocation, it is imperative that other signal patterns for aneuploids or unbalanced specimens have established cutoffs during the validation procedure. Overall, MECOM rearrangements are very heterogeneous and may be difficult to detect by conventional cytogenetics, which makes FISH a useful tool for their detection. Now let's review the general analysis guidelines that are specific to tricolor break-apart FISH probes. Again, analysts should avoid counting cells where the nuclei are too close together to determine their boundaries, as well as overlapping nuclei where all areas of both nuclei are not clearly visible. However, when there are single well-defined nuclei with nice, bright signals, You should count any cell that has a gap that is no greater than 2 signal widths as a fusion. This is true no matter if the gap is like this example, where there is a single red and green-blue side by side, or this example, where there is a single green and red-blue fusion, or this example, where there is a single blue and a red-green side by side. All of these cells should be analysed as 2RGB fusions. If you encounter cells that have a diffuse signal but are still attached to the others by a faint strand, they should also be analysed as 2RGB fusions. Examples shown here include a cell with a diffuse red signal attached to a green-blue fusion, a diffuse green attached to a red-blue fusion, or a diffuse blue signal attached to a red-green fusion. As with the dual-colour break-apart probes, it is imperative that all FISH technologists are trained to recognise these signal patterns in a similar manner to maintain the highest levels of concordance between analysts. The fourth specific sequence probe design strategy we will review is for translocation dual fusion probes. Different reciprocal chromosomal translocations are a common finding in cytogenetics, and they can be associated with a specific clinical indication, providing both diagnostic and prognostic information, as is the case with this AML1 ETO, also known as RUNX1, RUNX1T1 translocation dual fusion FISH probe kit that is available as one of OGT's FDA-cleared CytoCell FISH probe kits for AML and MDS. The chromomaps on the left depicts the probe set that consists of two 151 kilobase and 194 kilobase probes labelled in FITSI green on either side of the ETO, also known as RUNX1T1 gene, and two Texas red probes. One is a 156 kilobase probe centromeric to the AML1, also known as RUNX1 gene, including the CLIC6 gene, while the other is a 169 kilobase probe labelled in Texas red telomeric to AML1 gene, extending beyond the marker D21S1921. This translocation is commonly observed in patients with WHO subtype AML with recurrent genetic abnormalities. AML with translocation 821 Q21.3 Q22.1, which was previously known as AML FAB type M2. It is most commonly found in children and young adults and is a good prognostic indicator. In a normal sample, an analyst can expect to see the normal signal pattern of 2 red and 2 green signals, or 2R, 2G, which reflects 2 copies of normal genes for each of the targeted AML1 and ETO genes on chromosomes 8 and 21. However, in an abnormal cell, you will expect to see the ETO gene split on 8q21.3 and rearrange with a split AML1 gene on 21q22.1 that join together as 2 red-green signals, appearing as yellow or fusions, in addition to the normal red and green signals from one copy of the chromosome 8 and 21 that were not involved in the translocation. The final expected abnormal signal pattern will be 1 red, 1 green, and 2 fusions, or 1R, 1G, 2F. In addition to the reciprocal translocation 821 creating this typical signal pattern, variant translocations have also been reported for these genes, which could be cryptic and easily overlooked by G-banding. It is important to know variant translocation signal patterns are possible, as well as aneuploid signal patterns, and each laboratory should have their own established cutoffs for these potential patterns during the probe validation process. The analysis guidelines for the AML1 ETO translocation dual fusion FISH probe kit are similar to the ones previously discussed. Nuclei that are clumped should be avoided, as well as overlapped nuclei where all areas are not visible. An analyst should consider any nuclei that has a diffuse signal, such as this to be a 2 red, 2 green signal pattern. Additionally, when a gap is seen between two signals of the same colour that is no greater than 1 signal width, or where there is a faint strand connecting the two signals, as seen here, the analysts should consider this to be 2 red signals and 2 green signals. Remember, since variant patterns may be seen during analysis, it is important to have all analysts trained to read fusions and signal rearrangements in the same manner. When reading FISH probes, there are general guidelines that every technologist should follow. First, an analyst should assess the quality of the slide. Signals should appear bright, distinct, and easily evaluable, with greater than 50% of the cells containing a hybridized signal. There should not be excessive cellular background, haze, or clumped cells that may interfere with interpretation. If these qualities are not met, it may be necessary to rerun a new slide. Additionally, each lab should always follow their own analysis guidelines for slide interpretation. For OGT's FDA-cleared CytoCell FISH probe kits for AML and MDS, our IFU recommendations state that 2 analysts should analyse each sample and independently score 100 different nuclei for oncological specimens. The analysts should follow the guidelines discussed earlier in this presentation and only analyse intact, non-overlapped or crowded nuclei. Nuclei which are covered by cytoplasmic debris or a high degree of autofluorescence should be avoided. Analysts should also remember that signal intensity may vary, even on a single nucleus, so it may be necessary to switch between a dual or single bandpass filter, or adjust the focal plane to visualize all signals correctly. Similarly, there may be times when a third analyst is required. Each laboratory should establish its own requirements for when this may be necessary. For example, when 2 analyst results of abnormal signal patterns differ by a certain percentage. The flowchart on the right is a general guideline for interpretation and when to utilize a third analyst. Lastly, before interpreting results, any known probe cross-reactivities or limitations of the procedure should be taken into account. CytoCell includes probe cross-reactivities in both our IFUs and package inserts. Limitations and contraindications of the probe's use will only be found in our IFUs for our FDA-cleared probe kits. Prior to wrapping up today's session, we would like to quickly review some of OGT's many CytoCell product offerings. At OGT, we understand that using high-quality FISH probes decreases error rates and increases confidence in result interpretation. That is why we provide our customers with state-of-the-art DNA FISH probes. The majority of our catalogue FISH probe range is comprised of ready-to-use, directly labelled liquid probes in hybridization solution, which aids in minimising any potential mixing errors and streamlines processing. These include over 150 probes for various applications, including FDA-cleared FISH probe kits for AML and MDS, hematology, hematopathology, pathology, and constitutional cytogenetics. Moreover, through OGT's CytoCell Custom FISH Probe Design Service, or MyProbes, OGT can utilize our proprietary backclone collection to design a new probe that will meet your specific requirements. We can offer anything from a simple modification of an existing catalogue product to a completely new and innovative FISH probe. Please reach out to our team if you would like to be emailed the list of over 700 MyProbes options or discuss a new project. We would be happy to assist you. Furthermore, in order to assist our customers in their clinical and FISH research, CytoCell also provides a range of accessory products from DAPI, discussed earlier in this presentation, to a tissue pre-treatment kit or porcelain wash jars. Please let our team know if you would like a quote for any of these products. Before we close, We would also like to mention the many support options available to your team, both through our website and in person with our fish field application specialists. OGT believes that excellent customer service and support are the key to our success as a company. One way we offer support to our fish customers is through our online resources that can be found at www.ogt.com/US. Feel free to explore these offerings, which can be found by hovering above the resources header to produce the drop-down menu shown here. You can explore several topics covered in the fish resources and support section, from fish basics to fish tips and troubleshooting, or even watch our fish tutorial videos and read our scientific literature. These topics can be further expanded to explore things like what's wrong with my hematology fish, common fish FAQs, or general fish and tips, where our FAS team can give you pointers for every part of your fish protocol. Additionally, you can always reach out through the Contact Support link. The Contact Support link will connect with one of our fish field application scientists who are available to answer your questions, assist you with virtual troubleshooting, or perform on-site demos. Please feel free to reach out to them for any of your technical support needs. OGT would like to thank you for your time and attention throughout this presentation. We hope that you found this CytoCell University module useful. If you have any questions, please reach out to us at contact@ogt.com, and a local representative of OGT will connect with you shortly. Also, if you have any ideas for future presentations, or have any feedback, we would love to hear from you. Thank you.